My pleasure to welcome you all to AusCADASIL's first webinar, which is co-hosted by the Centre for Healthy Brain Aging at the University of New South Wales in Sydney and the John Hunter Hospital in Newcastle. So I am Perminder Sachdev. I'm Professor of Neuropsychiatry at UNSW and co-director of the Centre for Healthy Brain Aging. So before we begin, I'd like to acknowledge the Gadigal people and the Wabagal people who are the traditional custodians of the lands from where we are speaking, and I pay my respect to their elders past and present. So this seminar or webinar is one of a series of seminars that we're planning as part of our CADASIL initiative. Now, many of you who are attending this seminar probably are familiar with CADASIL. Some of you indeed may have the condition or know of somebody or have a family member who has the condition. Now, CADASIL is an acronym which actually stands for Cerebral Autosomal Dominant Arteriopathy with Subcortical Infarcts and Leukoencephalopathy. Now, that's a very long term, and fortunately, CADASIL is much more well known. But essentially, the long term means, firstly, it says it's autosomal dominant. That means it's a genetic disorder, that you're born with a specific gene and which has a mutation or a change. And this gene is called a Notch3 gene. Now, since genes come in pairs, one from each parent, autosomal dominant basically means that only if one of those two genes is abnormal, the disease will manifest. Now, it is cerebral arteriopathy, essentially because it affects the blood vessels in the brain, largely. And it says subcortical infarcts and leukoencephalopathy, essentially meaning that it produces small infarctions leading to strokes and also changes in the white matter of the brain. So really, that's what the terminology essentially means. Now, fortunately, CADASIL is rare. We do not have specific Australian data on CADASIL, but we estimate that about perhaps 1 in 100,000 people, 4 in 100,000 people have the disorder. From this, and of course, there are many figures actually have been put forward, but essentially we think that perhaps at least 500 people, but maybe as many as 1,250 people in Australia may have the condition. Now, in a recent study from the UK, they found that, in fact, 0.2% of the population, this was a random population that they had actually sampled, that is 1 in 500 had a variation of this gene, Notch3 gene, which is responsible for CADASIL. But since the prevalence is much lower than this, we do not fully understand the relationship between what genetic changes occur and how the disease becomes manifest in the body. But essentially what CADASIL does is it changes the small blood vessels in the brain, leading to small strokes, and often patients present with headaches, migrainous headaches, usually in their 40s and 50s, sometimes later. And often it ends up with disturbance in speech and motor function, changes in mood, and deficits in memory and other cognitive functions. And in fact, that's one area that has been less well studied and a particular focus for us because we work I'm a neuropsychiatrist, so my interest is in cognitive function more so. Of course, Professor Levi is a stroke neurologist, so he has more interest in stroke as well. Now, at present, there is no cure for this condition, and a lot more needs to be done to understand it and to recognize the variations and the early changes so that we can find effective treatment. Certainly, that's the goal. And I think that is where we would like to be at the end of maybe 5 to 10 years from now. So that is why this study, the first Australian cohort study of CADASIL was launched. It's part of an international effort. Actually, there is now a lot of excitement around the world in looking at CADASIL to see whether this is a disease that we can possibly alter the course of and perhaps eventually cure. So it's my privilege today to introduce two of our two speakers. First one is Professor Chris Levi. Some of you may already know Professor Levi, who's a leading stroke neurologist in Australia. He's based at the John Hunter Hospital and University of Newcastle. So he has extensive experience, both research and clinical experience in stroke, in CADASIL. He actually founded one of the first CADASIL clinics in Australia. And his other area is head injury, concussion. And he's focusing on new avenues for therapy and care. So he'll give you an overview of CADASIL research nationally and internationally. And the other speaker is Dr. Danit Sachs, who's a postdoctoral fellow with us at the Center for Healthy Brain Aging. She coordinates the AusCADASIL study. So Danit has a research background in neurodegeneration, dementia, and retinal biomarkers. And she's the main contact person for AusCADASIL, and she will provide you an update on the study. So following the presentations, there will be a live Q&A. So for the Q&A, if you would like to post any questions, do that in the chat function of this app, and we will pick those questions towards the end. Thank you again for attending, and I'll welcome Professor Levi for his presentation. Chris.
Thank you, Perminder. It's a great pleasure to be with you, and I just noted Patrick Clemenson's comment in the chat. I'm sorry to see you're not well, Patrick, and I'm very sorry to see that. But you are quite correct. This is a historic moment as a group of clinicians and community and patients and carers and others who are online. I really just wanted to make that point and acknowledge Patrick's comment. So I'll just put some slides up and share, but share the screen, but also just to acknowledge traditional custodians of the lands we're on and pay respects to elders past, present and emerging, and Aboriginal Torres Strait Islander people on the call here. So thank you, Perminder, for that introduction. And thanks, I acknowledge a number of our colleagues and collaborators on this joint venture are online as well, so can I thank them, and particularly Danit, who'll speak to you later, who's really coordinating this effort. I will just cover, as Perminder said, some key clinical points of interest and also what's going on in the research domain, but particularly in the AusCADASIL study. And as Perminder said, my background is in clinical stroke neurology, and I'm a clinical academic neurologist up at John Hunter in Newcastle. I also work in some administrative roles in the health district in research and innovation. But it's been an interesting journey for me over recent years. I did spend some time in UNSW and obviously very well aware of the efforts over many years of CHeBA in terms of healthy aging and dementia prevention. And it's been a great, I think, coming together of the neurology and neuropsychiatry fraternity now to focus on this important condition and see what we can do to try and advance knowledge and hopefully move towards better care and maybe a cure. So quickly, a quick timeline on CADASIL. We've been aware of this condition. Sorry, I should just say, Perminder will talk more about CHeBA and we can get into discussion, but I thought I should just acknowledge the activities of the group and its collaborative network. So really, that's the vision for the centre and groups, covering a whole range of aspects of neuropsychiatry, neurology, imaging, psychology, and importantly, the genetics and biomarkers area, which we'll talk about a little more. So, stroke is a very common condition, but familial types of stroke, although rare, are very important components of the mix of stroke. I mean, there are, as Perminder alluded to, a not insignificant number of sufferers from this condition. And the interesting thing about the familial types of stroke, they can be due to multiple effects of multiple genes of having small effects on the body. That's so-called polygenic stroke. And we know now there are over 50 genes that can have a small effect on the body and can contribute to anyone suffering a stroke, so-called common stroke. But we also know now there are a significant number of single genes that can have a mutation within them that have a very major effect on your risk of developing a stroke. And CADASIL is vastly the most common form of monogenic stroke or monogenic stroke, and the best understood. Quickly, a timeline to give you a bit of a feel for the, I guess, the research and our advancing understanding of CADASIL. It really was a condition that was first described, although not identified, back when I was a medical student, first year medicine, in fact, 1977 was the first report of what was then called a chronic familial vascular encephalopathy. Now it's a long-winded name for an unusual and previously unrecognized form of brain illness that really wasn't well understood. 1977. And for about 10 years, it was really relatively neglected until the late 80s, when people started to notice that there were families that began to suffer from what was then called multi-infarct dementia. And then with the genetic revolution, it became evident that there was a dominant gene involved in this condition. And in 1991, there was the description of families with this condition. But it wasn't until 1993 that there was the definition of the condition and the mapping of the gene to what we now recognise as the locus on chromosome 19 that Perminder mentioned to you, holds the Notch3 gene. A few years later, there was the recognition of a pathology in the small blood vessels that was first identified by electron microscopy, and then was recognised further. These granulophilic inclusion bodies around the small vessels. So there was evidence of something going wrong with the small vessels. And then in 96, the actual Notch3 gene was identified. So we moved then into the 2000s, and with this revolution in our ability to interrogate the genome, we began to understand where these mutations were in the Notch3 gene, and we recognised it's a large gene and there were many, many mutations across the gene, and we began to recognise other variants because we began to notice people with CADASIL symptoms and signs who didn't have a Notch3 mutation. And some of you will be aware of this. There is a more recently recognised version of CADASIL, so-called CADASIL2, which is a mutation in a different gene, HTRA1. And in recent years, the genetics has been further explored, and we now recognise that in CADASIL, in common CADASIL, I'll call it, or CADASIL1 for want of a better term, we now recognise across the entire genetic frame of the Notch3 gene, there are multiple variants, and some of those variants tend to result in earlier onset or more severe disease than others. And we're now moving to the point where we're beginning to model CADASIL in animals and understand how the pathophysiology, by that I mean how the gene affects proteins and how those proteins affect the structure and function of the small vessels, so-called translational genetics, translating from an understanding of what a mutation is to how it affects the proteins, how it affects the structure, how it affects the body. And that's really a critical step in understanding what you can do in terms of prevention and therapy. We don't have any effective therapies for the genetic abnormality itself, but that's really the next step. And I think, as Perminder said, we in Australia have really not as yet been major contributors to the international effort, but we're getting our act together now, and this is one of the important national efforts to actually start to contribute and start to understand where we go next. So Danit will talk probably a bit more about AusCADASIL. I'll cover it a little bit, just so you know, those of you who are involved and those of you who are thinking about what research opportunities might emerge in Australia. We'll tell you a little bit about that because it's our baby at the moment. Anyway, back to CADASIL. It's the most common, as I said, most common monogenetic form of stroke. Still relatively rare, but in terms of monogenic stroke, it is the common one. It is part of a whole wide spectrum of conditions that we do label as vascular cognitive impairment or vascular dementia, and we've known for a long time that there are some important modifying genes, particularly the gene called ApoE. I won't go into that. But in terms of vascular dementia, the important thing to recognise from this diagram I'm showing you, this schematic, is that there are risk factors that can be modified. It's difficult to modify your genes, of course. It's difficult to modify age, which is an important risk factor. But there are many risk factors that are manageable, treatable, both lifestyle-wise and with medication. I'll come back to that in terms of what the clinics that we're developing across the country will do. But there are significant preventable elements to vascular disease, prevention for stroke, therefore, and small vessel disease is out there, and we need to be mindful of it and implementing it, and that's one of the important aspects to what we're doing as a collective, not only doing research, but also blending that with good clinical care to help prevent as best we can the cognitive and stroke effects of these conditions. And you can see listed there lots of genes, lots of names. You see notch 3 there at the top of the list when it comes to genes causing stroke, but there are a number of other single gene or monogenic forms of stroke you can see listed there. So just to show you, it's a complex sort of field moving pretty quickly. But back to CADASIL. So I'll cover a little bit about the inheritance side. Perminder touched on it, but it is what we call a dominant gene, which means, as you probably remember, you get half your genome from one parent and half your genome from the other parent. And dominant genes, you only need one allele or one copy of the gene if it's abnormal to become expressed in the body, so to show the clinical features of what that abnormal gene and its protein product will produce. And because you inherit from each parent, you have a 50-50 chance of getting that gene, which I'll come to. I mentioned the NOTCH3 gene and the mutations across it. There are multiple mutations, but all the mutations tend to result in a single chemical substitution, a single amino acid shift from one favorable amino acid, which is one of the building blocks of the protein, to an unfavorable one. The unfavorable one is an amino acid called cysteine. And cysteine tends to be an attracting sort of amino acid and forms lots of bridges and bonds, and it disrupts the normal protein structures. And the abnormal protein structures around small blood vessels are where the pathology tends to lie, and I'll show you a little bit about that. And your small blood vessels are very important, obviously, not only for bringing oxygen and nutrients to the tissues, but also for reacting and maintaining good blood flow to the tissues. So I'll show you schematically a little bit more about that. But back to your genes. So there are these 4 amino acids. Cysteine's the one that tends to be substituted incorrectly in CADASIL, and it causes problems because of cross-linking and abnormal folding and configuration of proteins, and then their function becomes abnormal. So again, dominant, meaning one allele from one parent, can cause the disease. If you look at a recessive disease, the genes or the variants, the mutations are not as I guess you could say strongly affecting the tissue. So you need a double dose of a recessive allele to actually develop the disease. So they're less common because you've got to have both parents carrying that allele or that gene. But CADASIL, unfortunately, is a dominant, so it tends to flow through. So the inheritance pattern is that if, say, your father has an abnormal gene, you can see the little ticks are a normal Notch3 gene. The cross is an abnormal Notch3 gene. We know that by chance, probability is that abnormal gene will go to half of the offspring. It may well be by chance that if you have 4 children, none of them by chance will have received the abnormal gene, but the probability is roll the dice 50-50. So these 2 children here will carry the abnormal Notch3 gene, and in due course will have a high probability of developing the condition. So just to sort of orientate you to the blood vessel structure in the body, we have large blood vessels that come down to medium-sized blood vessels, to come down to small blood vessels, to come down to very small blood vessels, and then they form the veins, and the blood then circulates through the body from heart to artery to vein and back to heart. So CADASIL and this Notch3 mutation and its effect on that cysteine chemical and the proteins that cysteine is important in tends to affect these arterioles and these larger capillaries. So it's working down at this very small vessel end of the spectrum where your vessel has some muscle around it and also these other cells around it, which I won't go into the names of these, but there are all these cells that. So here's a cross-section of a vessel. The inside of the vessel is the white bit in the middle. There are all these cells around the vessel, and the abnormal protein affects a number of these cells and causes the vessel to effectively become narrow and less responsive to the normal physiological stimuli. And what that can do is produce in the brain Perminder alluded to this, 2 basic types of pathology. One is a small, deep stroke. You can see on this is an MRI scan of the brain showing you a little white dot here. And this little white dot's what we call in the trade a lacunar stroke. So people with CADASIL are more prone to these lacunar strokes. And if you get too many of these strokes, they can affect how your brain works. So they can affect your thinking, your speech, your coordination, your balance, your sometimes produce also other mental health side effects, depression, anxiety, apathy, fatigue. They have all sorts of direct and indirect effects. The other end of the spectrum is where the brain particularly what we call the subcortical parts of the brain, the white matter, is relatively starved of nutrients because of the disease in the small vessels. And here we see this pattern we call leukoariosis, or white matter disease. The outside of the brain, the cortex, is relatively spared. The white matter is more vulnerable to that subtle reduction in blood flow through because of the small vessel disease. And this is the leukoencephalopathy part of CADASIL. So this is the subcortical infarct part. This is the leukoencephalopathy part. So that's a classical sort of MRI picture. Common symptoms, again, Perminder alluded to this. The common symptoms are early in life, particularly even from teens, CADASIL sufferers tend to develop migraine, very common, over 70% of CADASIL sufferers. And most of the sufferers of migraine have migraine with aura. The vast majority have the funny vision that often precedes the migraine. In the general population, migraine with aura is less common than migraine without aura. So migraine with aura in the general population is only about 10 or 15% of migraine sufferers, but in CADASIL, it's up around 80%. The strokes, as I mentioned, particularly those small vessel lacunar strokes, the white matter change can slow people down cognitively, as can the strokes, and the cognitive impairment and dementia can develop. And as I mentioned, there are mental health effects of the changes in the brain. So the brain and the mind, as Perminder will tell you, neuropsychiatrist, as a neurologist, and my colleagues online will tell you, they are intimately linked. They're very impossible to separate. So you get significant mental health issues. There are some less common forms of CADASIL. In fact, I have only ever seen one case of CADASIL encephalopathy or CADASIL coma. But people with CADASIL can sometimes present quite acutely with confusion, being very sleepy or even unconscious, and they recover spontaneously from it. We don't understand why it happens. It's obviously got something to do with blood flow, but CADASIL encephalopathy is a less common form, and also seizures. Sometimes people with CADASIL can develop seizures, epileptic activity, and that's described. So I'll move on just to talk to you a little bit about what we're doing in the clinical space, and I'll talk a little bit about the research and leave Danit to talk a little more about that. So a number of my colleagues and I, for some years, have been thinking about, well, in many parts of medicine, it's become recognised that to give patients the best care, it's important to bring together teams of people who have special interests in a particular focus area. And we've known that for a long time. And stroke care is just one of those examples where stroke units have been formed. Now they make a difference to how people are treated and recover. But it came to me and a number of my colleagues that we hadn't really been doing this with CADASIL, and it's an important problem, and we didn't have ourselves organised in such a way to make the most of the knowledge that we have and concentrate it amongst a group of professionals who are able to keep up to speed and provide right diagnosis and best practice management. So the concept that is emerging across the country with this collaborative is that we establish, I guess you call them reference clinics, where we can make sure that patients get optimal clinical assessment, optimal brain imaging, optimal genetic testing, and the right genetic counselling in preparation for that, but also best practice management in terms of counselling, family planning, stroke prevention, dementia prevention, In fact, I heard an announceable today, Perminder, from your team. So congratulations on that. You might want to talk a bit about that, but and also management of symptoms such as fatigue, etc., which is a particular interest of mine. So this is what the clinics are doing, and we have collaborators in Sydney, Melbourne, Brisbane at the moment who are forming up. As Perminder kindly said, I have established the model clinic up here in Newcastle for our catchment. So now the research, and I'll finish up on this note. So over many years, and I'm sure same for Perminder and my other colleagues online, we've recognised that clinical medicine benefits from academic embedding and academic learning, growth in our knowledge and skills benefits from being close to clinical care. So AusCADASIL really is a blend, sorry about the spelling mistake there, a blend of clinical service delivery, looking at best practice care, but also trying to generate new knowledge. And we can only do that if the doctors and the nurses, the researchers, and the patients and carers all work together. So the concept here is we are putting together a collective. And a bit of a shout out again to Patrick, who's online, who's been a champion in terms of encouraging me and others to sort of do things in Australia to help advance CADASIL research, and we're often running, thanks to Perminder and his team, putting together a really very nice bid and having some funding to support this activity. So the objectives broadly of AusCADASIL are to make sure that we characterise the condition well, and we look at how the condition relates to how people feel, and we learn about what the spectrum of the condition actually looks like. So learn as much as we can about what we call the phenotype, that is how people are clinically. We also need to learn about what we call the endophenotype, which is not what you see, but what we see not see from the outside, but what you see when you investigate someone with tests, neuropsychology, imaging, blood tests, genetic tests. So I understand all those other deeper aspects of CADASIL, the neuropsychological profile, the MRI scan, the blood markers, and also we're interested in the eyes and how the small blood vessels in the eyes behave, because the eye is part of the brain, and it's a very neat way to look at small blood vessels by examining the eyes. We want to understand some of the predictors of poor health in CADASIL. Over my career, I've seen a wide spectrum of CADASIL. Some more fortunate people have very mild disease, often coming on later in life and not causing them much problems at all. But unfortunately, there are some people who have major problems from the disease, and we don't really understand why that is. So we need to look at the natural history, the longitudinal course of the condition. We need to understand more about how conventional risk factors can play a part in what we can, because we can do things about conventional risk factors. We need to understand more about the genetics. And most importantly for me, we need to be able to say when we get promising treatments, we need to be able to say to a group of CADASIL sufferers and their carers and supporters, we have something we would like to try. Are you willing to be involved in future research? So the cohort, the AusCADASIL cohort, really sets us up to look at treatments. That's really, for me, probably the most critical aim. And then finally, as Perminder mentioned in his discussion earlier, we want to be part of the international effort to cure this condition. So that's really where the broad aims are. It's a big collective across the country. of collaborators, neurologists, neuropsychiatrists, neuropsychologists, geneticists, basic scientists, genetic counsellors. There's even a cardiologist there. So, here you go. An enlightened cardiologist, Professor Kovacic, very good chap. So we've got a good team. And I'd just like really to finish by saying, take a message from me. There are many things we can do for CADASIL if we organise ourselves and provide you and the community with good access to high quality care. And the reference clinics across the country are a way of doing that. And then we have the Australian-based research that will contribute. And finally, just to thank all of you who have engaged in the study so far, or those of you who are supporting the study, but particularly thank Perminder and Danit who put this together tonight. And I wish you all well. And I think it's probably heartening for you to know that the research community and the clinical community are working together, and we're looking to try and build something here that will be a strong platform for future clinical care and knowledge generation in this condition. So thanks. I'll finish there, Perminder.
Thank you so much, Chris. That was wonderful. We have a lot of questions, so we'll come back to those once we heard Danit. So over to Danit.
So thanks for the introduction and thanks for the overview, Chris. That was really good to hear sort of how it goes from the genetics all the way through to the manifested disease. I think it'd be very useful for a lot of people to have that summary. So thank you. I just wanted to really introduce myself and give you all a little bit of an overview of where AusCADASIL is at and what is AusCADASIL for those that are new here. So my name is Danit. I'm a postdoctoral researcher at the Centre for Healthy Brain Aging, which is at UNSW. And my primary role is that I am the study coordinator for AusCADASIL. So I've met a lot of you already as part of the study and spoken to some of you on the phone. So it's great to see some familiar faces. So I really just wanted to draw your attention here to the different sites. So AusCADASIL is a multi-site study, as has been mentioned. So we are currently across 3 different states in Australia. So you've already met Perminder and Chris, who are the 2 New South Wales lead investigators, but the study also has a few other investigators from the different sites. You can see Amy Brotman, Jan Ho, Michael O'Sullivan, and Lynn Griffiths on the screen, and their different affiliations below. So each of them has a focus either on neurology or genetics, and they bring their expertise to the study, and we're very grateful for that. So it's been mentioned a little bit, but some of the outcomes that we're looking for in AusCADASIL and that we use to look at the different aspects of the manifested disease are some health and wellbeing questionnaires, which we tend to do online, but we do have the option to do in person if you'd like that extra support. We do neuroimaging to look at any brain changes that might have occurred. We do a comprehensive neuropsychology assessment, which is quite different to some of the other studies, which just look very briefly at cognition. But we look at really subtle changes in thinking and memory and also processing. We look at physical health. Has there been any change to gait, to strength, to BMI, blood pressure? We look at molecular biomarkers and genetics. And then we also look at ocular health in some of the sites. So at the moment, it's going to be Sydney, Melbourne, and Perth. Maybe we'll expand that. We'll see. So the reason why we have so many different outcomes is because, like Chris was saying, CADASIL doesn't present the same in everybody. So we want to know why. So why is there such a difference in the presentation, even within families? Is it because of the genetics? So we know that there's over 200 different variants within that Notch3 mutation. Is it because of that? Is it because of environmental factors? Is it lifestyle? Is it risk? Is it something that we can intervene? And is it something that we can prevent the risk or minimize the risk for further progression in the disease? So that's why we have such a comprehensive test battery in the study, just to really capture as much as we can about the disease. So with AusCADASIL specifically, where are we at now? So we have a few sites, but not all the sites are up and running, which a lot of you know. So Sydney and Newcastle are actively assessing. So thank you very much to Chris, to Beata, and to the 2 research assistants, Phoebe and Janelle, who've been doing the assessments over there. Melbourne is almost up and running. We'll be ready in a couple of weeks. So anybody who has approached the study from Melbourne, you'll hear from us soon to get the assessment started. And Brisbane and Perth are not quite as far along as we'd hoped, but we will be starting up there in a couple months. So we've now screened 53 participants and 18 controls, and we've had 28 participants and 8 controls come through the study for full assessment so far. But the target is 150 of both groups. So we're not quite there yet, but we are well on the way to getting our data. But that means I can't present any data to you today, unfortunately, but hopefully at the next webinar, there will be more to present. And as you know, we have the option for consent, the option for feedback to be sent to both yourselves and to any GPs or neurologists that you want to have this data. So that can really complement the clinical care and provide a service that others may not be able to access. So we're going to be starting follow-ups soon. So from April this year, we're going to be starting follow-ups. So this is going to be approximately a 2-hour visit where we'll do another neuropsych assessment, just an update on what we did previously, and also look at it if there's been any changes to your medical history, any new medical events, and how's your physical health as well. So a lot of you would have seen this, but I wanted to just share our research flyer. So this gives an overview of what the AusCADASIL study is, what are our assessments, and also who can participate. So we're not only looking for people with CADASIL that's been diagnosed, we're also looking for people with suspected CADASIL, so people that the neurologist has said may have CADASIL, and then we can do genetic testing and appropriate genetic counseling as well through the study for those individuals. And we're also looking for healthy controls. So these are people who don't have CADASIL. We can include family, but only if they've already been genetic tested themselves or if they're showing some symptoms. So aside from our flyer, I also just wanted to draw your attention to this brochure. So we've created this brochure to give an overview of just the basics about CADASIL. So what is CADASIL? How does it present? What are you likely to see with CADASIL? And the idea behind it is that it can be given to family members, to carers, supporters, friends, anybody that you might want to explain what CADASIL is, but you might not be able to find the words for it. We've got this brochure. So if you do want a copy of it, I'm happy to send you a PDF that you can use. And this can be for people who might participate in the study, but also just for family and friends if you want to inform them about what you're experiencing. So just really briefly, I wanted to mention global collaboration. So it's been said before that research really relies on international collaboration, and it relies on the sharing of both ideas, data, and support between research teams around the world. So I often get asked, why is there no CADASIL research? And it's not true. There is a lot of CADASIL research out there, but it's not necessarily at a stage yet that it can be published. So I just wanted to really briefly mention these 14 groups that we've had direct contact with around the world who are currently actively exploring CADASIL in various forms, either through cohort studies or registries, and are really actively looking into this condition and what they can do to help prevent, the progression in CADASIL, but also the development of manifested disease. So that's really all from me. I just wanted to give a brief overview. But if you haven't yet participated in AusCADASIL and would like to, please contact me at auscadasil@unsw.edu.au or just message me on the chat. I'll see it. And if you are part of a research group who isn't part of the Global CADASIL collaboration, please contact me as well. We'd like to speak to you. And I just wanted to say thank you so much to the participants who have already participated in AusCADASIL. We wouldn't be able to do the study if you weren't interested and you weren't contacting us and telling your friends about it and getting everyone involved. So thank you so much for being proactive about that and for getting involved. And I also just wanted to put up the QR code to our protocol paper. So that's the paper that Chris showed you before. This gives all the information about AusCADASIL, both in terms of what assessments we do, but also why do we do it and how does CADASIL fit in terms of the field of research, in terms of vascular dementia, and what can we learn from CADASIL. So if you've got your phone handy, you can scan the QR code. Otherwise, you can find the paper at cerebral, what's the CCCB? Cerebral Cognition, Circulation and Behavior. There we go. Which is a journal that this paper has been published in. And the paper is open access. So people often say to me that they can't access research because they need an institutional login or they need a subscription to the journal to be able to get access to that paper. But we have published this open access. So if you are interested, you can have a look, you can download it. And yeah, I'll hand over to Perminder to facilitate the Q&A, but thank you all for coming tonight.
Thank you so much, Danit. That was a very clear presentation. And of course, Chris, you are in line for a lot of questions. So we have everybody here. So perhaps, Chris, we can start off with CADASIL type 2? I think there are a couple of questions with interest in that. What are the differences? So other than the gene being different, how does it manifest differently? And there is one question about leg symptoms you might have seen. Yeah.
Yeah, look, they're very, very similar in terms of the phenotype and the imaging. The only clinical difference, to my knowledge, is that the type 2, this acute encephalopathy has not been described in it, so it's not part of the spectrum. But otherwise, pretty well identical in terms of the phenotype and how the patient looks and feels and what the doctors think of them. But it's a completely different gene, this HTRA gene. But obviously it's affecting a very similar sort of protein dynamics and microvascular function. It doesn't have those same deposits. Obviously, when I've talked about those little GOM deposits, those, if you look under the microscope at the vessels, they don't look exactly the same. They don't have those osmophilic little spots around the blood vessels. But I'm not aware of any other differences. Perminder?
No, but it's much more rare, of course, than CADASIL.
Yeah, that's right. So if it looks like CADASIL and the Notch3 genetic tests with the newer technologies that look at the whole gene are negative, then you should look for it. Yeah. But it is dominant. But it is dominant as well. So the pedigrees look the same.
Yeah. So, Danit, maybe you can tell us about the genes that are looked at when we send the blood. We send the blood to Professor Lynn Griffiths in QUT in Brisbane. Yeah. And she has a lot of experience with the genetics of CADASIL and related disorders. So can you tell us which genes are being looked at in that?
I actually don't have the list offhand, but I know we are looking at 15 targeted mutations that are similar to CADASIL. Heidi's actually on the call, so if you want to field that question, you're welcome to, but you don't have to.
Sure,
similar conditions.
Heidi, if you'd like to chip in.
Yes, I can't remember offhand exactly, but certainly HTRA1 is on that panel. And there's some other cerebral vascular disorder genes, for example, the collagen 4A1 and collagen 4A2, which can show overlapping symptoms with stroke disorder, the cerebrovascular disorders.
Yeah.
And there's a few other very, more kind of rare ones. HTRA1 mutations, we and others are finding that, yes, they're not as common as Notch3 mutations, but they may be half as common. There's certainly, there's quite a few mutations that we're finding. We can't always say whether they're definitely causal mutations at this stage. With the CADASIL, it's well understood that if you have that cysteine in the earlier part of the protein, then that's definitely a causal mutation, whereas these other ones, we find mutations, but if they haven't been described before, it can't be definitely said that that's what's causing the disease. So it's a little bit more difficult with some of those other genes at the moment.
Okay, so maybe
and the other genes we have on that panel are actually migraine-related genes, because a lot of people do actually present with migraines quite early on in the disorder. So we can exclude a migraine diagnosis from people with the Notch CADASIL diagnosis.
So can you maybe introduce yourself very briefly, Heidi, as well? We didn't introduce you yet.
Yes, I'm a postdoctoral researcher in Professor Lynn Griffiths' laboratory up in Brisbane. She's been doing diagnostic testing for various migraine-related disorders since 1997. We used to do like very targeted sequencing of certain parts of the Notch gene, but now with the developments of genomic sequencing, our diagnostic lab can screen all the coding part of the Notch3 gene as well as other genes at the same time to increase the diagnostic yield that you get out, when you are trying to get a genetic diagnosis. But we're also, our lab does research, and obviously there's a lot of people that come in with suspected CADASIL that don't have mutations in these known genes. So we're also interested in perhaps finding some other genes that might cause the similar symptoms.
Thank you, Heidi. So Chris, back to you. There is one question also related to CADASIL type 2, person with significant Parkinsonian symptoms and abnormal feelings in the legs. Is that something you come across?
Yeah.
Patients?
Yes, it is. So not uncommon to see this, what we used to call lower half Parkinsonism. So essentially the legs become more stiff and the gait becomes more shuffling, but the arms and the hands and the fingers are working okay. This is part of the spectrum of that subcortical white matter changes that we showed you on the MRI. And not uncommonly, the legs also get restless and you get that uncomfortable feeling in the legs. There's treatment for that in terms of the feelings, but the gait problem is difficult to manage. And we actually see it more in common stroke, to be honest with you, than we do in more advanced CADASIL, but you do see it in CADASIL.
Yeah, thank you. So people are also wondering about CADASIL coma, the mechanism perhaps of CADASIL coma.
Yeah, like the mechanism is not understood. So I've my clinical experience over 20 odd years, I've only ever seen one case, which tricked me because the person who came in with the CADASIL coma wasn't known to have CADASIL. Someone actually quite close to Patrick, I can see on the screen there. I didn't know Patrick at that stage, but no, it was certainly a diagnostic dilemma at the time. But we don't understand why it happens. But the good thing about it is it's reversible. It's spontaneously reversible without any long-term adverse effects. So the brain function returns to normal, and the person wakes up usually the maximum 48 hours. It's not really a TIA. No, I can see the question. No, it's quite different. It's a global shutdown of the brain where you really just go to sleep, and it's very alarming, very scary for everyone.
Wow.
Yeah.
No, thanks very much. There's a question about, I guess, spontaneous mutations as well, really, because I think that's people are wondering if you don't have a family history and yet someone has. So I suppose that's, again, Heidi, I don't know whether you want to speak to that or.
Sorry.
Yeah, go. Yeah, you go, Heidi. You know more than me.
Sorry, okay. Chris, you can come in.
No, Heidi, go.
No, sorry, no. I'll leave it to you, Christopher.
No. I don't know a lot about it other than it's. I mean, the de novo presentations are relatively uncommon in my clinical experience, but I don't know what the spontaneous mutation rate is.
Yes, I mean, obviously everyone is getting mutations all the time. We all get mutations. Every new person has hundreds of mutations, so it can happen, and it happens to all of us. But if it happens to hit in an important part of a gene, then the disease can, look like it manifests spontaneously. I'm not sure what the rate is, and sometimes that might be more a reflection of various mutations in different genes that happen to end up all together in one person. So it's maybe a sort of in between of the monogenic types of disease that Chris was talking about compared to the polygenic types of stroke. And so, yeah, I think, we work a lot in migraine related disorders, and there's again monogenic subtypes and polygenic, But I think a lot of there's much now accumulating evidence that there's these kind of middle of the road ones where they have a few mutations that mightn't cause the disease by themselves, but then when they're coupled with a couple of other mutations in other genes, then it manifests. So I think it's part of the variable penetrance or expressivity of diseases. So some people can have a mutation, or two people might have the same mutation, and the disease can actually come out in one person and not in the other. And we think that's a combination of other mutations in different genes, but it also might be what's called epigenetic factors as well that can contribute to that. So that means they're not DNA changes, but they're changes in whether the expression patterns of genes. They can there's other sort of changes that can be put on the DNA that make the gene switch on and off. So there's various things that can make a disease manifest or not. So it might have been hidden in the previous generation, and then just the combination of other mutations makes it manifest in that particular person.
Thanks very much. I guess we don't always test the parents or do genetic testing of the parents, or often they're not available for testing. Sometimes they may have mutations, but they were disease-free, so they weren't detected, or some parents might have passed away before they manifest the disease. So there are all sorts of reasons why you don't have the family history in a particular individual. It's a complex.
Linda, could I just mention, I saw the question from Kay about medication.
Sure.
And just to comment, there are some medications that help if this is that type of more restlessness, discomfort in the legs that and I assume they've tried what is called levodopa, which is the treatment for Parkinson's. Some helps for that, but there are other things as well. I'll just put some notes in the chat there. You might want to consider discussing those with your GP, Kate.
Or they could contact you, I guess, Chris, if that's the doctor could contact you, maybe.
They're happy to do that. Happy to do that. The other thing to say, I saw the note from Deborah just while I'm speaking, and you are quite right, Deborah. And Leanne's note pertinent. But it is difficult to educate and get sort of messages out, but essentially knowing that there are the reference clinics around and using telehealth, which is really, I think, the vehicle to begin discussions, is probably the way to go here. And there's a lot of work for us to do. We do work with the UK CADASIL group, who are an excellent group, and Hugh Marcus, who's a good colleague and friend of ours, came over to learn how to do telestroke from us in the Hunter, and then took it back to do it in the UK. So I think telestroke is the way to go here, or telehealth, I should say. So we will, as we evolve the network of clinics, look to respond to that need and try and do our best to get access and allow GPs to come into those consultations as well.
Thank you very much. I think we're running out of time now. We have to close. But there are several other questions, some of which we can probably try to address later or directly. If there is anything pressing, you can get in touch with Danit, and maybe she can direct you to somebody who's knowledgeable to answer your questions. I guess that may be the best.
See, from those questions, Danette, from Leanne, I'm happy to respond to those.
I think that's wonderful. Now, I think we should end thanking you all actually for your attendance today and for supporting this initiative of being volunteering, and in particular shout out again to Patrick, who has been involved from the beginning, who's been very enthusiastic in setting up this cohort as well. And we wish you good health, Patrick, and good recovery. And thank you once again. We hope to have another seminar sooner or later. We'll let you know. But meanwhile, Danit will be in touch with whoever wishes to participate in the cohort study. Thank you
Yes,
again.
and I just wanted to quickly mention while I've got everyone here, if you're not already a part of the CADASIL support group on Facebook, that's another one of Patrick's wonderful initiatives. But please do join. It's a really good space. Jamie's also on the call, who's a big part of that group. So please do join. It'll be somewhere that we can post things to you as well, but also that you can talk to each other and hear from other people with CADASIL. So if you're not a part of that, it's the CADASIL support group, Australia and New Zealand on Facebook. You can just Google and it should come up. Thank you everyone for coming. You'll hear from me in coming days. So if you do want to get involved in CADASIL research or you are awaiting a follow-up study, you'll hear from me. So I look forward to speaking to you then.